Oh, Nathan. Everybody comes. How are you all today? Good. Give me 1 second here. Anybody have any questions? All right, sorry, I'm having some technical issues here. All right, I think I'm good. For some reason, everything freezes on me once in a while. It looks like that's okay. Okay. All right, so nobody has any questions? Yes, it's fine to just watch if you're not feeling well. All right. All right. So Nathan, make sure you change your name there so that it's I keep track with it gives me a report afterwards, and if I see James Wood, I won't know who that is. Always make sure you use your, that's no problem. The computers always go, they default to whatever the person who created it is. So a lot of times you have to change that name right at the very beginning of class every time. All right, let's begin. All right, I'm all frozen again now. All right, so I'm going to have to restart here. I'll be back as quickly as I can. You guys just hold on a second, okay? Because I can't get anything to work. Ah, technology. Love it when it works, don't when it doesn't. Well, maybe there. All right, let's try this one more time. Maybe this will work. Okay. All right. Okay. So you guys learned last week about cell growth and division. This is so important for the information that's coming up in the next couple of weeks. I want to make sure that you really understand that we're all on the same page. I'm going to do a very fast review of cell growth and division. If you want more information, stop me. But if there's time after we get through this review, I want to talk about Punnett squares, which you'll start learning about in the lesson that has a due date of tomorrow. All right. So let me go here to a couple of little cartoons. Today we're going to learn about mitosis. I need your undivided attention. And did you guys know jokes about mitosis are not funny, especially when they're told twice? Why should you never talk about mitosis at a party? Because it's a divisive topic. And dude, mitosis starts in 5 minutes. I can't believe you're not condensed yet. Right? The condensing happens. And here's a way to remember mitosis. Start with one mic, you end up with 2 mics. Mitosis. All right, so cell size. Remember that cells can only get to be a certain size. They can't just keep growing and growing and growing. Most cells. Some cells obviously can get to be bigger than others. You've got nerve cells that go all the way from your bottom of your spinal cord to your toes. So that's a very long cell, but it's not big around. It's going to be very narrow. And of course, an egg cell gets to be a lot bigger than a skin cell does. So those are some exceptions to the cells. But in general, a cell can only get to be a certain size. So take a look at these dogs. These dogs are exactly the same size. But you can see that room A has a lot more volume. So the volume is the space inside. There's a lot more volume in that space. So even though the bigger room also has bigger walls, in other words, bigger surface area, the volume to surface area ratio is different in them. So kind of think of it this way. If your job was to feed this dog and carry out its waste products, how much easier would it be to get to the dog in this little room than it would be to get to the dog in the big room? That's because it's a long way from the walls to the dog in the big room. Now, the same thing applies to a cell. It's a long way from the membrane of a cell to the inside of the cell, where everything has to get the nutrients, get the oxygen, get the building materials that it needs, and then get rid of the waste products that it needs. So if a cell can't get too big, we have to have a solution. Our solution is cell division. That's where you take one cell and divide it and it becomes 2 cells, and then they can become 4 and they can become 8 and so on. All right. It's a couple of terms we need to make sure that we understand: chromatin, chromatid, and chromosome. So chromatin, that is the DNA that's all loose inside the nucleus when it's not getting ready to divide. So it looks a little bit like a bowl of spaghetti in this picture. If it's not dividing, it's this wad of chromatin, and it looks a little bit like this ball of spaghetti. Now, it's more complicated than that. I'll talk about in a second. If it is not dividing, it is not called a chromosome. It's still DNA, but it's not called a chromosome. It's called chromatin. Once it starts dividing and getting ready to multiply like this, it begins to wrap around these balls, these silver balls called histones. It begins wrapping around them. Then they wrap up into these sticks that are called chromatids. Now, when it's wrapped into a chromatid, it's called a chromatid, but it's also called a chromosome. I know that's tricky. So when it's wrapped into a chromatid like this is called a chromosome, but it's also called a chromatid. When you have just one, it's a chromosome. But when there are 2, you also have a chromosome. It's a little bit confusing with that. A little bit like learning another language. Just try to keep in mind the important ones are chromatin before it starts condensing and wrapping into the X shape in order to multiply. And then chromosome can be either 1 or 2 of the chromatids. There's another cartoon. Mitosis, it happens every second. Yeah, and it does. So stop and think about that for a minute. Stop and think about that this process is happening in every cell in your body, every minute of the day, your entire life long. There are 2 million cells that are being replaced in your body every second. 2 million, 2 million, 2 million every second. Since there are 86,400 seconds in a day, that means that there are 172.8 billion cells that are going through this cell cycle every day in your body, just to keep you functioning, just to keep you alive. Now, these are all images, all pictures, drawings of the cell cycle. So you can see they have some similarity, but there's also some differences. They've all got a longer interphase. So the dark green right here is interphase. This whole, all these 3 colors, that's interphase. This one right here, basically everything that's not mitosis is called interphase. And they're all drawn in a circle to show that this is a process that has no beginning and no end. Now, what we have to remember is that there's always another layer of learning whenever you got something new that you're coming into. Like if you take a look at this one right here, there's pro-metaphase. We didn't learn about that one. We learned about prophase, metaphase, anaphase, telophase, and cytokinesis. What's pro-metaphase? Well, it's another layer. It's just a little bit more complex than we need for our class for this purposes. But basically what we have is a longer interphase and then a shorter mitosis. That's the M right there, mitosis. And there's 4 main parts of mitosis. There is prophase, metaphase, anaphase, and telophase. Now, be sure that you're paying attention to all this, right? So it's important that we understand what these different phases are. I remember the cell phase, the cell cycle, right? That, sorry, I mean for that to go there. I remember that by remembering IPMAT, IPMAT, just remembering. There's a lot of different ways online that you can find little sayings to help you remember those, but keeping an eye on them, learning what order they go in is important. Now, from a bird's eye view, meaning what's the whole thing about anyway? The whole thing, the whole purpose of the cell cycle is DNA, right? That's what this is all about. The cell has to make an identical copy of DNA so that in the end, there are two identical copies. So that when the cell splits into two in cytokinesis, each of those new cells has a duplicate copy of DNA. Why, if DNA matters so much, why don't other things matter? What about the organelles? What about the mitochondria and the ribosomes and the lysosomes and the Golgi apparatus? What about them? None of this have I said anything about duplicating the mitochondria. They're pretty important. They make energy for the cell. Why do you think that they're not important? Because they're not. Any thoughts? Basically, if you have the DNA, you have everything that you need. The DNA has the code to make more of everything. The DNA has the code to make the proteins that the mitochondria are made of. It has the code to make more ribosomes, to make more endoplasmic reticulum. So if you, by chance, on one side of this cell, if all the mitochondria went onto this side and none of them went onto this side, it wouldn't matter. The DNA would just make more. All right. Now, you got all this DNA in the nucleus and you have to duplicate it. That happens in that long phase called mitosis. You can kind of think of mitosis as being, interphase. You can kind of think of interphase as being the waiting room. It's what is going on before cell division occurs, but a lot happens in that waiting room. There isn't just sitting around happening. So interphase is the period in between cell divisions. Now there's phases of interphase. So we've got our G1, that G stands for growth. So growth phase 1, that's just where normal growth is happening. Then notice this checkpoint right here. That's kind of an important one. Then we've got our S phase. S stands for synthesis. That's where DNA is being made. There's also some other proteins that are being replicated in this that are necessary for cell division. Then you've got your G2, your second growth phase. Final protein synthesis happening here for proteins that are needed in mitosis. Then we have another checkpoint. Then prophase, metaphase, anaphase, telophase. Now we've got another checkpoint right here in the metaphase right there. It's not on this image, but there's one there. We've got these checkpoints. They check for errors all the way along. If they find an error, they have to fix it. If they can't fix it, there's a lot of things that can happen, but the one that doesn't happen is the cell continues into the rest of the cell cycle. If there's an error that can't be fixed, it stops the cell from dividing. Now, mitosis right here, this is the big one. That's really where the whole thing, it's what the whole thing is about. The whole cell is preparing for mitosis. These 4 parts, PMAT, prophase, metaphase, anaphase, telophase. Let's look at each one of these. Now here you're looking at the whole cell. This blue right here is the cell. So here's your rough endoplasmic reticulum. Here's your mitochondria. These would be a couple of maybe ribosomes in there. But you can also see that you have your spindle fibers forming here, these little strings. You can kind of think of them coming off of the centrioles as a little bit like fishing poles with wire attached to them, with fishing line attached to them. So in prophase, the way you can tell it's prophase is you see one set of X's. You just got one of these, the X's in the nucleus. That's the only time that they're just loose in the nucleus. So here is an image that shows you, a little animation that shows you. As soon as they attach, now prophase is over. Then comes metaphase, where the attached spindle fibers pull those chromosomes into the middle of the cell, and they form a plate in the center of the cell. So this is what it looks like right here in metaphase. Now this is a 3-dimensional plate. It's not just a line, it's a 3D plate. Once they're lined up in the middle, now metaphase is over. Next is anaphase. This is where that fishing line, those spindle fibers start to shorten, like they're being reeled in, like a fisherman reeling in his catch. And it pulls half of each chromosome toward the opposite ends of the cell. See these little microtubules on the other side of the centrioles? They start pushing against the side of the cell to change its shape. They make it into an oval shape instead of a round one, so that it's got more structure when it begins to pinch off in cytokinesis. Here you can see an anaphase, they're being pulled apart. Metaphase is for middle, they're in the middle of the cell. Anaphase, they're being pulled apart. And then finally, we have telophase. So in telophase, the cell is still one cell, but look at the DNA. There's two sets of it, here and here. The nuclear envelope is beginning to form again around it, and the chromosomes start unwinding, so they look like a bowl of spaghetti again. The nucleolus begins to form again. That's this round structure right there that makes ribosomes. And these are still pushing. The cell keeps getting longer. This is what telophase looks like. So those spindle fibers are shrinking back into the centromeres, or the, sorry. There you go. And then they're pushing, they're elongating the cell right there. And then you can begin to see the nuclear membrane reforming. Now keep in mind that at the same time that telophase is happening, cytokinesis is happening too. So the cell starts pinching in at this place called the cleavage furrow, and a ring of microfilaments starts constricting at the center until finally it pinches that cell off into two separate cells. Now, all those organelles begin assembling using instructions from the DNA. So if more mitochondria is needed, the DNA sends the instructions, and we end up at the end with two separate cells. Now here, you can see the whole thing in 3D. So let's watch this. Remember I said it's all in 3 dimensions. It's not a flat cell. It's kind of hard to picture it. So here we have prophase. Once they're lined up in the middle, metaphase. Anaphase are being pulled apart. And then in telophase, the nuclear membrane reforms, there's cytokinesis. But remember that the whole thing is in 3 dimensions. So as we turn this, you can see nuclear membrane broke down, prophase, metaphase, that's the plate, anaphase, they're being pulled apart, and then telophase. 2 cells where we just had 1 before. All right, now I want to do a Kahoot to see how well you understand this. For this Kahoot, I don't want you to use your real name because I want you to feel anonymous about this. I want you to use this to check yourself to see how well you understand this. So this is not a competition. This is you figuring out whether or not you understand all this or not. After this Kahoot's over, if you didn't do well, then I want you to go back to the lessons about mitosis and read them again. Because like I said, they are important for what we are going to be continuing to learn in the next couple of weeks. All right, so www.kahoot.it. Don't use your real name. This is just you checking your own knowledge here. It's a short one. There's only 14 questions. Your game pin is 649-2008. Original name. I said don't use your original name. There you go. 649-2008. I got half of you in. Who knows? Six 649-2008. All right, going once. 1, 2, 3, 4, 5, 6, 7, 8. 1, 2, 5, 6, 7. Still missing one of you. I'm going to go ahead and get it started. You can always join using this game pin. It's always down at the bottom. All right. So see how well you do. True or false? Mitosis creates two genetically identical cells. True. Genetically identical means that it has exactly the same DNA. That is true. Excellent job. Put the four main phases of mitosis in correct order. Click on what comes first. Prophase, metaphase, anaphase, telophase. All right, 4 of the 7 of you didn't get that right. So make sure that you learn what order they go in. Remember, that's why we're here, learning where we need to brush up. What is the period between cell division called? This is like the waiting room. This is interphase. Interphase. The intermediate step. Which phase of mitosis does this image show? All right, that is prophase. I'm going to show you guys why. You have X's. You have them. The nuclear membrane is still there. In metaphase, you're not going to see the nuclear membrane at all. I wish I could make that a little bit bigger. I can't. A little bit bigger. The X's are going to be in a line along there in a plate, not scattered throughout. All right, makes sense. In which phase do duplicated chromosomes become visible and a spindle begins to form? When can you begin to see those duplicated chromosomes? That is in prophase. The spindle fiber begins to form right at the beginning of prophase. Telophase is at the very end. So the chromosomes become invisible again. They begin unwinding and becoming just that bowl of spaghetti again in telophase. In which phase do chromosomes separate and move toward opposite ends of the cell? Anaphase for apart. They are being moved apart. Type your answer. If a cell has 24 chromosomes, how many chromosomes will each daughter cell have after mitosis? All right, the correct answer is 24. Yeah, they'll be identical. They'll be identical with one another. I would have accepted any of those. All right, so 4 of you guys got that. Remember, in mitosis, we have identical daughter cells. So if the cell had 5, the daughter cells would have 5. If the cell had 100, the daughter cells would have 100. They would be identical to what it was in the beginning. Which process completes cell division by splitting one cell into 2? That is cytokinesis. Cytokinesis, where we end up with 2 cells. Cytokinesis happens with telophase. Which phase does this show? That is metaphase. We can tell it's not prophase because we don't see any nuclear membrane. Nuclear membrane is still a little bit there in prophase, but it's gone in metaphase. And we have a straight line, a flat plate of those chromosomes. A few more questions. In what phase does the nuclear envelope reform around chromosomes and the nucleolus is visible? Telophase. It is telophase. That's the end. That's the last phase before cytokinesis. Which phase of mitosis does this image show? Anaphase. They're being pulled apart in anaphase. Double points. How is cytokinesis in plant cells different than in animal cells? Plant cells form a cell wall. Yep, they form a cell wall. Animals don't have that. What phase of mitosis? All right, that's telophase. That's the end. We've got 2 nuclei. These are reforming their nuclear membranes. The chromosomes are beginning to unwind to become just chromatin again. That is telophase. And last question. How confident are you in your knowledge of mitosis? Not confident yet. I need to study more and ask for help. Somewhat confident, but I need to study more. I'm confident in my knowledge and ready for a test. I'm so confident I can teach my peers about the topic. All right, somewhat confident, but need to study more. All right, good. I hope that was helpful. These are, like I said, important reviews. Oh, let's go ahead. I'll cancel. We'll go ahead and see, even though we don't know. Lettuce in 3rd place, bruh in 2nd place, and in 1st place, we had science with a smile. All right, great job, everybody. All right, so we do have time now to go into talking a little bit about what we're going to be talking about this week. So you guys started with him last week. This is Gregor Mendel. He's the scientist who started the whole thing. We call him the father of modern genetics because he's the one who first started researching about how genetics leads to the traits that it does. Now, we'll learn as we go through this next chapter that a lot of what he believed wasn't true. It was later proven to be wrong, but a lot of it was right. And what's really important about Mendel is not just that he got things started, but he really used very good lab procedures. He took meticulous notes. He did a lot of going over and over and over the same experiment over and over again. So very, very, very big sample size. And he used a great plant. Peas were the perfect choice for this kind of study because they've got a lot of characteristics that are contrasting with one another. You know, round or wrinkled seeds, yellow or green seeds, you know, inflated or constricted pod shapes. A lot of very contrasting ones. They have a pretty short life cycle. Doesn't take very long from when you plant one, you know, germinate or fertilize one until you have a full plant. They produce a lot of offspring. They can pretty easily be handled in a laboratory. So you can intentionally pollinate them because they reproduce sexually. Now, there aren't a lot of things that are like that. Think about a human. We're not easily handled in a lab. We don't have a lot of offspring. We don't have a short life cycle. So by using something like peas, he was able to get a lot of data in a really short amount of time. So you guys will start learning about these really cool tools called Punnett squares tomorrow. I know they can be a little bit intimidating, but once you know how to use them, they are very useful. So we're going to spend time today practicing how to use these Punnett squares. This is what it is. It's just a tool to predict the possible genotypes for the offspring of 2 known parents. What do I mean by genotype? We're predicting a genotype. What's that mean? Somebody has to know. We have genotype and phenotype. What's a genotype? Yes, good, Robbie. What the genes of an offspring would be. Yeah, genotype is the genes. It's what the actual genes that you inherit are going to be. Phenotype is the physical manifestation of those genes. So let's just review a couple of important terms before we go on. What do we call an allele whose trait always shows up when it's present? Chrissy.
The dominant allele.
Yeah, that's dominant. That's going to be a dominant allele. What do we call one whose trait only shows up when the dominant one is not there? Isabel.
Recessive.
Yeah, that's going to be recessive. So you can think of the dominant one as being bossy and the recessive one as being small. Now notice that they both have, they're both the same letter. So if we've got like green seed color is dominant over yellow, we wouldn't use a G and a Y. We would use a big G and a little g. So the little g would stand for yellow. With a big B is going to, the big allele is always going to be dominant. It's going to be bossy. And if it's there, the little one or the recessive one is going to hide. So here we have again, dominant and recessive. So we're going to use uppercase letters for dominant, lowercase of the same letter for the recessive ones. Here's genotype and phenotype again. The genotype is the actual genes that you get. The phenotype is the observable form. So you can see them right here. Tall, short, green, wrinkled. Those are things you can observe, things you can see. You can't observe TT, GGT, you know, those. Those are the genes that they actually get. So here again, you can see the genotypes. Those are the alleles or the individual genes that they get. The phenotypes are the physical manifestation of those. Now, let's talk about a Punnett square. Let's just make a couple of them. So what we do when we're making a Punnett square is we start at the top and drop down and put the letter in the box. Big B is going to drop down to both of those. This big B here is going to drop down here. Big B, big B. Now those are all done. Now these are both little b, so we're going to carry those across. Little b carries across. This one is little b carries across. That's it. We just made a Punnett square. Now we can tell if we cross this dominant right here, two dominant ones, that would be called homozygous dominant, with this one, which is also homozygous, but it's homozygous recessive. I've lost my pointer there. Homozygous recessive because they're little b's. If we cross those two organisms, we would get 100% one dominant, one recessive allele, or heterozygous offspring. All right, now let's continue on. Let's do this again. So if we drop down from the top, we're going to drop down a little t right there. Now we're going to drop down another little t. We just drop it into the box again and again. Now we're going to carry one over and one over. All right. Does anybody not see how I'm making that? Text me privately if you want to. You got to drop them down and carry them over. Same way. Now we can tell if we cross this homozygous there we go. This homozygous recessive, two little t's, with this heterozygous right here, big T, little t, we're going to get two that are heterozygous and two that are homozygous recessive. That's what. Now, understand that if these two parents have 4 offspring, they're not going to get one of each. You have this same odds every single time. So every single time we would cross these two, we would have a 50-50 chance of getting this one and a 50-50 chance of getting this one. Now, let's see what we can do with it. We know how to make this tool. Let's see what we can do with this tool. So let's suppose that we have here the possible outcomes of a cross between black and brown feather color in a particular bird. And we're told that black is dominant over brown. Now, if we're told what the parents are, we are given the parents' genotypes. Big B, crossed with little b. Homozygous dominant, crossed with homozygous recessive. All right. Now, what color is the, well, let's see here. Let me put them in. All right, I'm gonna have to do this way. So we've got a homozygous dominant right here, and we've got a homozygous recessive right here. All right, let me take those off and draw them in this way. Okay, do you see why? Everybody see how I'm doing that? That's this one right here, right there. I put them both on the Punnett square. They are both homozygous. Okay, now we're going to fill in the squares. So we fill in the squares. Dropping the letters on the top into each square. Okay, big B, big B, big B, big B. That's all that parent had to give. They had all dominant alleles. Now we're going to carry it over from the side. Little b, little b, little b, little b. The order doesn't matter in the boxes, but almost always putting the uppercase first is what they do. You could have written this little b, big B, but it's almost always done this way. All right, now let's look at our results. What are our results going to show? If big B is the dominant allele for black feathers, and little b is the recessive allele for brown feathers, we can make predictions about the outcomes. So here's our results. Our phenotype, that's what the colors of the feathers are. 100% of the offspring is going to have black feathers because 100% of the offspring is getting at least one dominant allele. So we're going to have 0% of the feathers being brown. That's our phenotype. Now, when we look down at genotype, that's right here. Genotype is what the actual genes are. 100% of them are going to be big B, little b. All right. Let's do another one. T, big T, is the dominant allele for plant tallness. What is the recessive allele going to be? Chrissy.
The plant being short, that would be the
Yep, but what's the allele going to be? If the allele for dominant is a big T, what's the allele for shortness going to be? Chrissy?
little t.
Yeah, it's going to be a little t. That is going to be our allele, our recessive allele for a plant that is short. We're going to drop them down. See, these two came from this one here. We dropped them down. These two below it came from this one here. Then we're going to bring it over from the side. We're going to carry that big T over and carry the little t over. And this is what we're going to have. Little t, little t. Now, our results. What do you think? What is the phenotype going to be? If big T is the dominant allele for being tall, what percent of these are going to be tall? Or how, what fraction? Yeah, you're right, Robbie, 75%. 75%. So we've got this one here. That's going to be tall because there's at least one dominant allele. This is going to be tall, and this is going to be tall, at least one dominant allele. So we are going to have our phenotype 75% tall, 25% short. Now, genotype is going to be the genes that they got. That's our letters that we're going to use. With our letters, we're not going to get, there's not 75% of anything with genotype. We've got one of these, we've got two of these right there, and then we've got one of these. These two are the same. So our genotype is going to be 1 to 2 to 1. 1 big T, little t, 1 big T, 2 big T, little t, and 1 little t, little t. All right, now let's use this in an actual real case. Here's flies. These are Drosophila melanogaster. We'll call them fruit flies. In these fruit flies, red eye color is dominant. So it's going to be a big R is going to give us red eye color. Brown eye color is recessive. We're not going to use a little b, we're going to use a little r for the recessive. If we crossed this heterozygous with this homozygous recessive fruit fly, what percent of their offspring would be expected to have brown eyes? All right, we put that into a Punnett square. The red-eyed parent, this one right here, is heterozygous. The brown-eyed one is homozygous recessive. So we'll put the parents in first. We put the big R from here, right there. That one came from there, and this one will go right there. It's going to be the little r. This one is going to go here, and this one's going to go here. So we're going to have a little r in both of those. All right, now we can answer the question. Our question was, what percent of their offspring would be expected to have brown eyes? Remember, brown eyes is recessive. In order to get brown eyes, you have to have two little r's. We fill this in. See where I got that? Dropped it down, carried it over, big R, little r, little r, little r, big R, little r, little r, little r. Anybody not see how I got that? All right, so what percent would be expected to have brown eyes? Yep, good, Isabel, 50%. These right here. The only way to get brown eyes is to have two of the recessive alleles. So what percent would be expected to have the red eyes? Also 50%. Now, again, I'm going to say, if these flies had 4 offspring, that does not mean that 2 of them would have red eyes and 2 of them would have brown eyes. It means every single time there's a cross between these two, you would have a 50-50 chance of getting red eyes or brown eyes every time. All right, let's do another one here now. We're going to do this backwards. In this cross, what are the genotypes of the parents? So we can carry it back, if you will. So we know this one right here is going to be a big B. And we know this one right here is going to be a big B. We know this one right here could have gotten this big B from here, but there's one more big B, so it's going to be from there. Now, this one got its big B from there. So what does this have to be right here? Has to be a little b. Does that make sense then if this one got the big B and the big B there? Yes. So the correct answer is the homozygous dominant and heterozygous are the genotypes of the parents. All right, so you can go both ways with this. Figure out the offspring, figure out the parents. Now, male, female, that's a little tricky because male and female do not have dominant genes. If you have one X chromosome and one Y chromosome, you're male. If you have 2 X chromosomes, you're female. So let's fill out this Punnett square. Let's complete this one. We're going to drop this X down and carry this X over, and we're going to get 2 X's right there. We're going to drop this X down and carry this X over. We're going to get 2 X's. Drop this X down, carry this Y over. We get an XY. Drop this X down, carry this Y over. We get XY. So what are the odds of getting a female child in any given cross? You got it. What are the odds of getting a male child? Every time the odds are 50-50. Every time there's a pregnancy, there's a chance that that pregnancy will result in a male 50% of the time and a female 50% of the time. I'm going to say it again. That does not mean if these parents have 4 children, they get 2 girls and 2 boys. Right? It means every time there's a 50-50 chance. All right. Is this an accurate Punnett square? Why not? What should it be? What should go here? Yeah, big T, little t. What should go here? Yeah, big T, little t. Are these 2 accurate? Yes, those 2 are accurate. How about this? Is this one accurate? Getting some yeses and some nos. Yes, this is accurate because this big G is both of these. The little g came down from all of them, and this big G went there. So that is accurate. How about this one? Yes, that one is accurate. Excellent. Excellent. All right. Let me jump ahead here. Okay. So which one of these shows the dominant allele? What letter is the dominant allele in this case? Yeah, the big Y is going to be the dominant allele. These two are heterozygous because they have one of each. These two right here, what do we call them? They're not heterozygous because they don't have one of each. Homozygous, but you need more. Homozygous what? Yeah, you got it. They are homozygous recessive, right? If they had two big Ys, then it would be homozygous dominant. And if I said to you, circle the dominant allele, you would just circle one of the big Ys. You would circle that. That's the allele. This is not an allele. That's two alleles. All right. Which one of these shows the dominant allele? Which one? Well, let me rephrase that. Which of these offspring has at least one dominant allele? Yeah, all of them. So which one would result in an offspring that showed the recessive trait? Yeah, you're right. None of them. None of them would show the recessive trait. All right. Let's see here. I'm gonna do this one. Okay, let's do a real one. If parent A is homozygous dominant for dimples, parent A is at the top, homozygous dominant for dimples, what alleles is it going to have? What's going to go across the top? Homozygous dominant for dimples. We're going to use D for dimples, though. The parent is A. Yeah, you got it. So we're going to have the two uppercase. If parent B is homozygous recessive for no dimples, what is parent D going to have for their alleles? Yep, two little Ds. Excellent. All right, what is the expected phenotype of the offspring? That's a genotype. You guys give me genotype, and you're right. They're all going to be big D, little d. What is their phenotype? Yeah, that we're going to have 100% heterozygous. 100% are going to have dimples. Yep, excellent. All right. Take a look at this one now. What percent of the offspring can be expected to be homozygous dominant in this one? 25% are homozygous dominant. What percent are heterozygous? 50. And what percent are homozygous recessive? 25. What do these parents have to be? Not. Yeah, they have to, but let's back it up and see. They have to both be big T, little t. This big T came from there. This one came from there. All right, this is a big T here. So this one has to have come from over there, meaning this one is a little t. And the only way to get this homozygous recessive is to have another little t. So they have to be, both of them, big T, little t. All right, all right, do another one here. In pea plants, Green seeds are dominant. Now look at that. They give it a big Y, and they do that sometimes. Sometimes you'll see it like that. Don't just assume then that it's a G just because it is dominant. Usually it is, but sometimes they'll do it this way. So if in pea plants, green seeds are dominant, big Y, and yellow seeds are recessive, little y, the following Punnett square shows a cross for seed color. Is there a chance of getting yellow seeds? No, there's not. We're going to get 100% green because 100% of them have at least one dominant allele. In fact, 100% of them have just one dominant allele. Very good. All right, let's do a couple more here. Which of the following has the little h genotype? A, B, C, or D? Right over here. Yeah, it's going to be B. It's going to be just this one. The only one has to get it from both parents. So right there and right there. Very good. That is the correct answer. Let me see if there's. Okay, just to make things fun, we're also going to learn about these dihybrid crosses. Don't get too panicky when you see these. We don't really do a whole lot with these, but I want you to be aware of them, and you'll read about them in the reading. So when we're just comparing one characteristic, we do a monohybrid cross. When we compare two at the same time, we call that a dihybrid cross. And my sister works in she works in science. She works at a lab at the University of Iowa, and she told me that she frequently has to do trihybrid crosses with 3 traits. She said it's kind of a nightmare. You have a computer help you with that kind of work. Yeah. All right. So the way we would fill this out is, again, we're going to drop the things from above down into the boxes. So we're going to drop that one down, down, down. You guys see how I did that? The big S and the big Y. I'm going to do the same thing in with the big S and the little y. Then the little s and the big Y, and then the little s and the little y. Now I just have to do the same thing over. I have to carry it over. The big S and the little and the big Y is going to go over. The big S and the little y. The little s and the big Y, and the little s and the little y. All right? Simple as that. Yeah. Now, so if I said to you, what are the odds of getting a plant that was homozygous recessive in both traits? Our odds are 1 in 16. It would be this one right here. 1 out of the 16 possibilities are homozygous recessive in both traits. All right, I'm going to end us right there. Anybody have any questions? All right. I hope you enjoy Punnett squares. I find them to be a lot of fun to do. So hopefully this helped to prepare you for the lesson. People freak out about them a little bit, but I think that a little advanced work helps. All right. Have a terrific week, everybody. I'll see you next time. Bye.